My name is Donald Martin, and I'm a professor of nanotechnology at the University of Grenoble Alpes in France. My teaching is in the Faculty of Pharmacy, where we teach nanobiotechnologies and aspects of nanotechnology for medicine and pharmacy. My research is in the laboratory TIMSI, which is located near the hospital, and in that laboratory I have a team of scientists. We work on aspects of nanotechnology related to biotechnology, and those aspects are particularly related to proteins that we extract from animals and bacteria, from cells of animals and bacterial, to put into lipid membranes to create structures, and we term this biological engineering, to be able to make structures that can mimic the function of cell membrane transport. And that's the basic building block for the type of nanobiotechnology that myself and my team do. And the application of that technology is in terms of recreating transport processes. Why? Because the human cells, animal cells, plant cells, all transport ions and different molecules across the cell membrane, their cell membrane. In the process of transporting ions across the cell membrane, there's an accompanying gradient of ions that's produced that generates a very large electrochemical potential. The advantage for biotechnology is to harness that electrochemical potential that we can recreate in an artificial system that is based and built upon the biological engineering of real live components, real live proteins, into lipid membranes to recreate artificial cells, either on a flat surface, which is no longer a cell, of course, then becomes a biosensor or a fuel cell, or we can turn that into a cellular-based system of artificial cells, which can have a function, not exactly life, but it can have a function of transport that mimics cell function. The advantage of doing that is to, as I said, was to harness the immense electrochemical energy that's created from ion transport. And as an example, one of the systems we're working with now is a system of millions of connected vesicles, all connected with a transport protein, and as a system this can produce very, very large gradients of ions that we're harnessing into producing electrical power. We're harnessing into producing ways to influence cells that are in close proximity, because cells in the body naturally communicate through molecules and ions that are secreted. So by recreating an artificial system based upon biological components, we can build biotechnological systems that start with the ability to create a compatible system with the body. And in that way, we're aiming with this work to make types of biotechnology that can improve implanted medical devices, for example, that can produce very small but highly productive energy sources for the body. We can also think to produce very, very sensitive biosensing systems that are based upon the natural proteins that are in the cell membrane. So that's a very brief overview of what my work is about, and the presentation that I'll be making at the MAGI conference is to describe some of those systems and that detail. So thank you.
Very interesting.